The other thing that happened 10 years ago was that I was asked by the Department of Health and NIHR to lead a team to improve our research into rare disease and into genetics generally in the health service. It was obvious that we were doing less research. We'd always, through my early years in clinical practice, been involved in research projects alongside our counselling of families. And that was because it was quite easy to sort of send a sample off to a friend to be tested. But as the regulations on patient samples became more onerous, although entirely justified, it made it more and more difficult to actually do disease projects that involved rare conditions, which meant you had to collect samples across many different centres. So we created a team called the Collaborative Group on Genetics in Healthcare, and we took responsibility for the clinical research network in the National Institute of Health Research for genetics to try and increase the number of people with genetic diseases taking part in research. And it was very successful, and we became usually the top recruiting team, despite the fact genetics is quite a small specialty. Two of the things that we're particularly proud of, and I have to say this was very much dependent on my colleague Jill Borthwick, who was the manager of this programme, was to actually get everyone in the country to sign up to a project called the DDD project, which was nationally funded at the Sanger Centre in Cambridge. And the idea was to use a technology called exome sequencing. Instead of using the whole genome, you look at the 2% that where the coding genes, the 20,000 or so genes that we can read in the genome, can be looked at instead of the whole picture. It's a little easier to handle. And doing exome trios, where you look at mum and dad and the affected child. And we did that on 13,000 families collected across every genetics centre in the UK. So everyone signed up thanks to our collaborative project. And the exciting result of that was that more than a third of them got a diagnosis. These were people who'd been through the whole system and we couldn't crack it. And here we were getting a diagnosis thanks to these new technologies. So that health gain has fed into the development of the 100,000 Genomes project.